Good.
Okay, then I'll get started. So my name is Isabelle. I'm currently from Northern California. Cancer research has been something that I've just really enjoyed throughout high school. And so today I'm going to be presenting on one of the literature review projects that I've conducted, and it's titled The Role of Epithelial Cell Extrusion in Carcinoma. Okay, so did you know that up to 90% of all cancer types in the world are classified as carcinomas? And these are essentially cancers that occur or at least start in a tissue called the epithelium. And you can kind of see on the diagram on the left that the epithelium is really like a barrier. It separates the green part or the lumen, as well as the stroma or the underlying tissue, which you can see in red. And all of the lining in our body, including the skin, is considered an epithelium. And therefore, it's super important for the tissue to undergo a constant cycle of cell death and cell division in order to maintain its barrier against external damage, like you can see in the bacterial infection that's going on over here. And so when we look at something that goes wrong in the epithelium, a lot of carcinomas can develop and become lethal once they metastasize or they spread throughout the body. And in most cases, metastasis follows this general model that you can see here. We have a giant primary tumor, and cancer cells will detach from this tumor and spread through either the bloodstream or the lymphatic systems to other areas of the body. However, scientists have also discovered an alternative mechanism, which is cell extrusion. So you can see based on the direction that the cell is extruded or squeezed out of the epithelium, so either upward into the lumen or downward into the basal area or the underlying tissue, the cancer cells could have a chance to invade or be removed. And this extrusion mechanism was what I really wanted to study in depth. And throughout my project, I wanted to focus on the mechanical and chemical aspects, as well as to propose a few drugs for plausible treatment. So how exactly does extrusion work, and why is direction, related to it? So depending on direction, epithelial extrusion is classified as either apical or basal. And apical extrusion is usually the default or the normal direction. So old cells or any damaged cells are extruded outward into the empty space. And this can effectively eliminate any cancer cells through a process known as the epithelial defense against cancer, or EDAC. However, sometimes cells will go the wrong direction, which is underlying tissue, and they go downward basally. And this really allows the cancer cells to bypass a lot of the death signals, and they'll be able to invade and metastasize throughout the body. And so how exactly does this extrusion mechanism work? The actual mechanical forces are generated by something known as actin-myosin contraction. And just like the name suggests, we have two proteins, actin and myosin. They accumulate in the cells and they sort of pull the neighboring cells to form a ring. And it's sort of like a drawstring pouch. They keep contracting until the extruded cell is pushed out and it seamlessly closes the gap that's left behind in order to maintain the epithelial barrier. And so obviously there's going to be some sort of chemical component that's involved, and a lot of signaling pathways will exist to tell the mechanical process for extrusion when to start and when to start extruding. And one of the common pathways used for this is the calcium wave. So you can see in this picture, there's the center extruding cell, and it's going to be releasing a whole wave of calcium to its surrounding cells, which you can see in the hexagon. And this generates a lot of pulsatile force, which you can see by the kPa on the bottom boxes. And this is what generates enough force for that actin-myosin contraction to start contracting. And meanwhile, a second pathway known as the S1P, S1P2 pathway is what is assembling the actual actin-myosin components and also giving the signal for the cell to start contracting. So you can see that cell extrusion is a super highly coordinated process. And so obviously when the coordination goes awry, it can lead to things like carcinogenesis or the formation of cancer. And more often than not, this is due to mutations or genetic errors. For example, the KRAS mutation will lead to a faulty active myosin formation, and it will also dysregulate. Is key to regulating the ring formation and contraction. And so these mistakes will cause the cells to extrude in the wrong direction, and it will allow things like pancreatic cancer, lung cancer, colon cancer to invade the body. And what's super interesting is that basal extrusion in the wrong direction can actually grant cells a sort of new superpower called plasticity. And so cells that are incorrectly extruded will lose all of their characteristics and de-differentiate back into a stem-like form. And this allows them to be very drug resistant, as well as to migrate to other parts of the body. And so while I was researching, I noticed that a lot of skin, lung, and colon cancers all involve organs that function as a barrier of exchange. And so I was super curious to see whether or not they had the same extrusion mechanism as well. And there were several pieces of evidence that pointed towards my theory or hypothesis. And so I first found that there were increased rates of basal extrusion in the skin of zebrafish, and this actually caused a lot of tumor-like growths to form. There was also a similar phenomenon in the intestinal villi, or the projections of mice intestines, and a lot of defective extrusion caused blebbing and outgrowths, and the extrusion mechanism used was also similar to actin-myosin contraction. And this occurred even in the lungs, where a lot of bronchial cells underwent a similar stem-like transition that you can see in cancer. And even on a genetic level, cell extrusion and lung and colon cancers are highly correlated. And so obviously, based on those observations, I wanted to research more into the treatment of carcinomas. And so currently, the most used drug is chloroquine, which is used to restore the expression of S1P. And this basically allows cells to restore their normal apical extrusion in the right direction. But obviously, it's not perfect. There are results that vary based on the cancer type. A lot of drug resistance develops. Currently, scientists have also looked at FAK inhibitors. This essentially inhibits FAK, which is a survival signal, and that prevents a lot of the transformed cancer cells from bypassing death. And obviously, again, there are other side effects. We're not able to selectively target cells, so this means that a lot of healthy epithelial cells are going to be killed as well, and this leads to things like poor gut barrier function or nausea and vomiting. And so based on this, I wanted to sort of overcome these shortcomings. And so I looked more into a lot of drugs that could be repurposed. I found that wound healing and cell extrusion are very similar. So a lot of skin repair drugs such as alprostol or trimabutene malate could be repurposing candidates. And both of them actually activate the same MEK-ERK pathway, which will restore the apical extrusion and obviously activate the EDAC response against cancer, and also allow tumors to grow to a large enough size so that we can be able to remove them through surgery. And so obviously my conclusion is that epithelial cell extrusion is implicated in multiple different carcinomas, and it's able to do this through reverting to the basal direction for extrusion, impairing the actin-myosin contraction, and also dysregulating pathways. And obviously this is super important as not just a bridge between cell division and death, but also an alternative mechanism for cancer metastasis. And it might also be able to show us some drug candidates or explain other epithelial-based diseases. And these are my references, and thank you so much. Thank you so much to my mentor, especially Janice, for guiding me throughout the process, as well as to the Polygence team. And thank you all for listening.
Thanks so much, Isabelle. Great job. I put in the chat, I really love the drawstring analogy. I thought it made it really clear to me how it works. So that was great. We have a question from a fellow participant in the chat. Are most cancers considered carcinomas because most of our organs have epithelium and most cancers manifest on organ epithelia?
Yeah, that's actually very true. Well, statistically, you can see, obviously, most cancers do originate from the epithelium. And also because the epithelium, again, covers so much of our body. It's pretty much everywhere. And also because it's an easier place for us to manifest cancer, because the epithelium is where you're constantly having cells grow, as well as divide. And I think I froze a little bit over there.
I heard you though, yeah.
Okay. And obviously that gives more room for mistakes to occur, mutations to occur, and that will also lead to cancer.
Super interesting. That answered my question too, because I was wondering why so like such a high percentage was coming from that type of cell. So really cool. Thanks so much, Isabelle. Great job.